Cornell Cooperative Extension of Orange County presents 4-H Incubation and Embryology Online.
Welcome to Incubation and Embryology, our third episode, and today is called Candling the Eggs. But before we get started, I need to introduce my special guest today. Franken Beans!
So while we were away, let's see what our eggs were doing. On day 17, our tongue begins to form and our embryo separates from the yolk sac. Day 16, the formation of our reproductive organs and our gizzard forms.
15!
On day 15, the little features of our legs and wings begin to form. What happens with 14 days to go?
Candling!
Awesome. So on day 14, the comb begins to grow, our egg tooth begins to form, and voluntary movement occurs.
Welcome to 4-H Incubation and Embryology. Today is lesson 3, candling. We're going to find out what candling means, things we are looking for, as well as things that we don't quite want to see when candling. So let's start. Candling eggs. Candling is the process of looking inside of an egg without cracking it. We do this to see if the chick is developing. A bright light is used to candle the egg. Things we are looking for when candling. Blood vessels. This is the embryo growing, and eventually it will be developing very quickly, and it will fill up the entire egg. So when we see these blood vessels, it's a key. Air cell. We will be marking the air cell to see how it changes during our embryo's development. The eye. The eye is the largest and darkest body part formed at day 7 of candling. So those are our 3 things that we are majorly looking for when candling to know if our chicks are developing. Now, bad eggs, why we identify them and why it's so important. So the first thing that we did find when we candled our eggs is a detached air cell. One of our Pekin duck eggs had a detached air cell. This means that this egg is not viable. No cells divided, so no embryo was ever formed. We take this egg out of the incubator immediately so that it doesn't take up room for our viable eggs, as well as it doesn't begin to cook as we process it at a 99.5 degree temperature. Incomplete blood ring. An incomplete blood ring occurs when the embryo begins to develop, but the blood vessels never completely form to the embryo. This egg is also not viable. Although we did go back and check our duck eggs again, on the second time that we candled, it still was not viable. This told us that it was definitely an incomplete blood ring and we needed to remove it from our incubator. Ruptured air cell or scrambled. With one of our chicken eggs, when the air cell was broken, as an egg sits in the incubator, as I said before, it will begin to cook because it is at 99.5 degree. This egg must be removed immediately because it is not viable. This means that an embryo never formed and that the cells never divided. So this egg is actually infertile. So let's go over our checklist before we begin class. Have we washed our hands? Have we made sure that our incubators work? Have we added water? Is the temperature still 99.5 degrees to 100 degrees Fahrenheit? Okay. Have we gone over our rules? First rule is we do nothing, but it's candling day and we have to do something, so let's make sure we wash our hands.
First, we wash our hands. I see a dark spot.
You see the dark spot?
I see a dark spot
so
right
ready?
there.
So the first thing we're gonna do is we're gonna trace the air cell,
Yeah,
okay?
we know.
Or the air sac, very good. And the next thing we're gonna do is we're gonna look for the dark spot.
Oh, I found it.
Do you see it? So don't touch. This right here,
I know.
that.
Is that Molden?
Yep, this right here is the eye. It's the largest developed part of our chick embryo.
Oh,
So
wait,
is
that's a chick's eye?
it's a chick?
It's
yeah.
moving. It's moving.
It is.
Why
So
is it
is this
moving?
a yes, a no, or a maybe egg?
Yes.
Yes.
But why is it moving?
Because the baby's growing and developing.
Wait, it's moving?
Mm-hmm.
I thought it was still developing.
It's
and
still
sitting
developing. It can still move. All right, Evie.
there. I see something.
All right, but what's the first thing we do?
Circle the air sac.
All right, so circling our air sac is so important because as our chick develops, we're going to be able to see less and less light, and the air sac is going to move as the chick takes up more of the egg. So now what are we going to look for?
The eye.
Yep, that's the shell. So look over here.
Ow, you just bumped into me.
What do you think that is?
The eye.
The eye. So is this a yes or no?
Yes.
All right, so what's the first thing we do?
Circle the air sac.
Very good. So we're going to circle the air sac.
You always. I see something.
You do.
Really I saw it.
All right, wait. Let's make sure we check this. Because our air sac is going to move as our eggs develop. So let's see if we could find that eye.
Wait.
You see a dark spot?
Yeah, I see a shadow. Me too.
All right, so is that a speckle or is that a chick? Let's try to wake our baby up.
Pick it up? Why?
Yeah, because we want him to move closer to the shell so we can see him. Chicky. Oh,
We found him.
right here.
Right there. We found him.
You see him? Right here. I'll get him.
It's so dark.
I know.
See it.
So is this a yes or a no?
Yes. I found him. Again?
Yep. Okay.
it was
So
so
this
good.
one, can you see this round area at the bottom? I'm going to trace it. This is called the air sac, and that's what we're shining the light through. Okay, so some of our breeds take a little bit longer to see, but let's see what we can see in this egg. Can we see the yolk?
No.
Is it the darker spot?
Oh yeah, I see it.
See it
No,
moving?
that's the baby.
So is the baby the yolk or is the baby on top of the yolk?
On top of the yolk.
Very good. All right, so this other egg, we're gonna call this a maybe, okay?
Yeah. Another maybe.
Okay. This is our Pekin duck egg. So they take a little longer to develop, so we're gonna see what we can see today. But what are we gonna do first?
Circle the air sac.
Perfect. All right.
What?
Now, what we're gonna look for is we're gonna look for the eye. Do you
Oh, I
guys see it?
Yeah, I see it.
So this is the eye, and these are the veins that flow through to feed our embryo as it grows.
Are we gonna circle that?
So we're just gonna circle the eye, but that's a good question. But do you want me to draw some of the veins?
Yeah.
Awesome.
But why
So
is it
this
not moving?
So it is moving a little bit. It's just it takes a little slower
Oh, yeah.
than the chick eggs. All right. So this is a Pekin duck, and is it a yes or a no?
Yes. Yes. Ooh.
Okay. So this is our second
Okay.
duck egg, and we're gonna circle the
Air sac.
air sac. Okay.
I said that at the same time.
All right. So we're gonna check out and see what we see.
sac. I said
All right, so right here we can see there's kind of a tiny dark spot, and we can see some veins, but we can also see that the yolk looks kind of broken, right?
Let me see. Wait, I can see.
Do you see how it's a different shape?
Yeah.
All right, so this is going to be a maybe egg. We're going to check it in another 2 days, okay?
Yeah.
All right, so this is a maybe egg.
On the next episode, Frank and Beans. Science experiment.
See you guys soon.